Botulism, Wikipedia article audio. Botulism is a rare and potentially fatal illness caused by a toxin produced by the bacterium Clostridium botulinum. The disease begins with weakness, blurred vision, feeling tired, and trouble speaking. This may then be followed by weakness of the arms, chest muscles, and legs. The disease does not usually affect consciousness or cause a fever. Botulism can be spread in several different ways. The bacterial spores which cause it are common in both soil and water. They produce the botulinum toxin when exposed to low oxygen levels and certain temperatures. Foodborne botulism happens when food containing the toxin is eaten. Infant botulism happens when the bacteria develops in the intestines and releases the toxin. This typically only occurs in children less than 6 months old, as protective mechanisms develop after that time. Wound botulism is found most often among those who inject street drugs. In this situation, spores enter a wound, and in the absence of oxygen, release the toxin. It is not passed directly between people. The diagnosis is confirmed by finding the toxin or bacteria in the person in question. Signs and symptoms. Infant botulism. Prevention is primarily by proper food preparation. The toxin, though not the organism, is destroyed by heating it to more than 85 degrees Celsius for longer than 5 minutes. Honey can contain the organism, and for this reason, honey should not be fed to children under 12 months. Treatment is with an antitoxin. In those who lose their ability to breathe on their own, mechanical ventilation may be necessary for months. Antibiotics may be used for wound botulism. Death occurs in 5 to 10% of people. Botulism also affects many other animals. The word is from Latin, botulus, meaning sausage. Early descriptions of botulism date from at least as far back as 1793 in Germany. The muscle weakness of botulism characteristically starts in the muscles supplied by the cranial nerves, a group of 12 nerves that control eye movements, the facial muscles and the muscles controlling chewing and swallowing. Double vision, drooping of both eyelids, loss of facial expression and swallowing problems may therefore occur. In addition to affecting the voluntary muscles, it can also cause disruptions in the autonomic nervous system. This is experienced as a dry mouth and throat, postural hypotension, and eventually constipation. Some of the toxins also precipitate nausea, vomiting, and difficulty with talking. The weakness then spreads to the arms and legs. Severe botulism leads to reduced movement of the muscles of respiration, and hence problems with gas exchange. This may be experienced as dyspnea, but when severe can lead to respiratory failure, due to the buildup of unexhaled carbon dioxide and its resultant depressant effect on the brain. This may lead to respiratory compromise and death if untreated. Clinicians frequently think of the symptoms of botulism in terms of a classic triad: bulbar palsy and descending paralysis, lack of fever, and clear senses and mental status. Infant botulism was first recognized in 1976, and is the most common form of botulism in the United States. Infants are susceptible to infant botulism in the first year of life, with more than 90% of cases occurring in infants younger than 6 months. Infant botulism results from the ingestion of the C. botulinum spores, and subsequent colonization of the small intestine. The infant gut may be colonized when the composition of the intestinal microflora is insufficient to competitively inhibit the growth of C. botulinum and levels of bile acids are lower than later in life. Complications. The growth of the spores releases botulinum toxin, which is then absorbed into the bloodstream and taken throughout the body, causing paralysis by blocking the release of acetylcholine at the neuromuscular junction. Typical symptoms of infant botulism include constipation, lethargy, weakness, difficulty feeding and an altered cry, often progressing to a complete descending flaccid paralysis. Although constipation is usually the first symptom of infant botulism, it is commonly overlooked. Honey is a known dietary reservoir of C. botulinum spores and has been linked to infant botulism. For this reason, honey is not recommended for infants less than 1 year of age. Most cases of infant botulism, however, are thought to be caused by acquiring the spores from the natural environment. Clostridium botulinum is a ubiquitous soil-dwelling bacterium. Many infant botulism patients have been demonstrated to live near a construction site or an area of soil disturbance. Cause. Infant botulism has been reported in 49 of 50 U.S. states, and cases have been recognized in 26 countries on 5 continents. Infant botulism has no long-term side effects, but can be complicated by hospital-acquired infections. Botulism can result in death due to respiratory failure. However, in the past 50 years, the proportion of patients with botulism who die has fallen from about 50% to 7% due to improved supportive care. A patient with severe botulism may require mechanical ventilation as well as intensive medical and nursing care, sometimes for several months. The person may require rehabilitation therapy after leaving the hospital. Colonization of the gut. Clostridium botulinum is an anaerobic, gram-positive, spore-forming rod. Botulinum toxin is one of the most powerful known toxins. About 1 microgram is lethal to humans when inhaled. It acts by blocking nerve function through inhibition of the excitatory neurotransmitter acetylcholine's release from the presynaptic membrane of neuromuscular junctions in the somatic nervous system. This causes paralysis. Advanced botulism can cause respiratory failure by paralyzing the muscles of the chest. This can progress to respiratory arrest. Furthermore, acetylcholine release from the presynaptic membranes of muscarinic nerve synapses is blocked. This can lead to a variety of autonomic signs and symptoms described above. Food. In all cases, illness is caused by the botulinum toxin produced by the bacterium C. botulinum in anaerobic conditions and not by the bacterium itself. The pattern of damage occurs because the toxin affects nerves that fire at a higher frequency first. Wound. Mechanisms of entry into the human body for botulinum toxin are described below. The most common form in Western countries is infant botulism. This occurs in infants who are colonized with the bacterium in the small intestine during the early stages of their lives. The bacterium then produces the toxin, which is absorbed into the bloodstream. The consumption of honey during the first year of life has been identified as a risk factor for infant botulism. It is a factor in 1/5 of all cases. The adult form of infant botulism is termed adult intestinal toxemia, and is exceedingly rare. Inhalation. Toxin that is produced by the bacterium within containers of food that have been improperly preserved is the most common cause of foodborne botulism. Fish that has been pickled without the salinity or acidity of brine that contains acetic acid and high sodium levels, as well as smoked fish stored at too high a temperature, presents a risk, as does improperly canned food. Foodborne botulism results from contaminated food in which C. botulinum spores have been allowed to germinate in low oxygen conditions. This typically occurs in home canned food substances and fermented uncooked dishes. Given that multiple people often consume food from the same source, it is common for more than a single person to be affected simultaneously. Symptoms usually appear 12–36 hours after eating, but can also appear within 2 hours to 10 days. Wound botulism results from the contamination of a wound with the bacteria, which then secrete the toxin into the bloodstream. This has become more common in intravenous drug users since the 1990s, especially people using black tar heroin and those injecting heroin into the skin rather than the veins. Wound botulism accounts for 29% of cases. Isolated cases of botulism have been described after inhalation by laboratory workers. Botulism has occurred after cosmetic use of inappropriate strengths of Botox. Injection. The toxin is the protein botulinum toxin produced under anaerobic conditions by the bacterium Clostridium botulinum. Mechanism. Clostridium botulinum is a large anaerobic gram-positive bacillus that forms subterminal endospores. 1971 Bon Vivant botulism case. On July 2, 1971, the U.S. Food and Drug Administration released a public warning after learning that a New York man had died and his wife had become seriously ill due to botulism after eating a can of Bon Vivant Vichyssoise soup. Between March 31 and April 6, 1977, 59 individuals developed type B botulism. All ill persons had eaten at the same Mexican restaurant in Pontiac, Michigan, and all had consumed a hot sauce made with improperly home-canned jalapeno peppers, either by adding it to their food, or by eating a nacho that had had hot sauce used in its preparation. The full clinical spectrum of type B botulism was documented. In April 1994, the largest outbreak of botulism in the United States since 1978 occurred in El Paso, Texas. 30 persons were affected, 4 required mechanical ventilation. All ate food from a Greek restaurant. The attack rate among persons who ate a potato-based dip was 86% compared with 6% among persons who did not eat the dip. A 13.8, 95% confidence interval, 7.6 25.1. The attack rate among persons who ate an eggplant-based dip was 67% compared with 13% among persons who did not. Botulism toxin type A was detected from patients and in both DIPs. Toxin formation resulted from holding aluminum foil-wrapped baked potatoes at room temperature, apparently for several days, before they were used in the DIPs. Food handlers should be informed of the potential hazards caused by holding foil-wrapped potatoes at ambient temperatures after cooking. In 2002, 14 Alaskans ate mudok from a beached whale, resulting in 8 of them developing botulism, with 2 of the affected requiring mechanical ventilation. Beginning in late June 2007, 8 people contracted botulism poisoning by eating canned food products produced by Castleberry's Food Company in its Augusta, Georgia plant. It was later identified that the Castleberry's plant had serious production problems on a specific line of retorts that had underprocessed the cans of food. These issues included broken cooking alarms, leaking water valves and inaccurate temperature devices, all the result of poor management of the company. All of the victims were hospitalized and placed on mechanical ventilation. The Castleberry's Food Company outbreak was the first instance of botulism in commercial canned foods in the United States in over 30 years. 1 person died, 21 cases were confirmed, and 10 more were suspected in Lancaster, Ohio, when a botulism outbreak occurred after a church potluck in April 2015. The suspected source was a salad made from home-canned potatoes. A botulism outbreak occurred in Northern California in May 2017 after 10 people consumed nacho cheese dip served at a gas station in Sacramento County. 1 man died as a result of the outbreak. There are 8 serological varieties of the bacterium denoted by the letters A to H. The toxin from all of these acts in the same way and produces similar symptoms. The motor nerve endings are prevented from releasing acetylcholine, causing flaccid paralysis and symptoms of blurred vision, ptosis, nausea, vomiting, diarrhea or constipation, cramps, and respiratory difficulty. Botulinum toxin is broken into 8 neurotoxins, D, E, F, and G, which are antigenically and serologically distinct but structurally similar. Human botulism is caused mainly by types A, B, E, and F. Types C and D cause toxicity only in other animals. In October 2013, scientists released news of the discovery of type H, the first new botulism neurotoxin found in 40 years. However, further studies showed type H to be a chimeric toxin composed of parts of types F and A. Diagnosis, prevention, vaccine, treatment. Some types produce a characteristic putrefactive smell and digest meat. These are said to be proteolytic. Type E and some types of B, C, D, and F are non-proteolytic and can go undetected because there is no strong odor associated with them. When the bacteria are under stress, they develop spores, which are inert. Their natural habitats are in the soil, in the silt that comprises the bottom sediment of streams, lakes, and coastal waters and ocean, while some types are natural inhabitants of the intestinal tracts of mammals, and are present in their excreta. The spores can survive in their inert form for many years. Toxin is produced by the bacteria when environmental conditions are favorable for the spores to replicate and grow, but the gene that encodes for the toxin protein is actually carried by a virus or phage that infects the bacteria. Unfortunately, little is known about the natural factors that control phage infection and replication within the bacteria. The spores require warm temperatures, a protein source, an anaerobic environment, and moisture in order to become active and produce toxin. In the wild, decomposing vegetation and invertebrates combined with warm temperatures can provide ideal conditions for the botulism bacteria to activate and produce toxin that may affect feeding birds and other animals. Spores are not killed by boiling, but botulism is uncommon because special, rarely obtained conditions are necessary for botulinum toxin production from C. botulinum spores, including an anaerobic, low salt, low acid, low sugar environment at ambient temperatures. Botulinum inhibits the release within the nervous system of acetylcholine, a neurotransmitter, responsible for communication between motor neurons and muscle cells. All forms of botulism lead to paralysis that typically starts with the muscles of the face and then spreads towards the limbs. In severe forms, botulism leads to paralysis of the breathing muscles and causes respiratory failure. In light of this life-threatening complication, all suspected cases of botulism are treated as medical emergencies, and public health officials are usually involved to identify the source and take steps to prevent further cases from occurring. Botulinum toxin A, C, and E cleave the SNAP25, ultimately leading to paralysis. For botulism in babies, diagnosis should be made on signs and symptoms. Confirmation of the diagnosis is made by testing of a stool or enema specimen with the mouse bioassay. Antitoxin. Physicians may consider diagnosing botulism if the patient's history and physical examination suggest botulism. However, these clues are often not enough to allow a diagnosis. Other diseases such as Guillain-Barré syndrome, stroke, and myasthenia gravis can appear similar to botulism, and special tests may be needed to exclude these other conditions. These tests may include a brain scan, cerebrospinal fluid examination, nerve conduction test, and an hydrophonium chloride test for myasthenia gravis. A definite diagnosis can be made if botulinum toxin is identified in the food, stomach, or intestinal contents, vomit or feces. The toxin is occasionally found in the blood in peracute cases. Botulinum toxin can be detected by a variety of techniques, including enzyme-linked immunosorbent assays, electrochemiluminescent tests, and mouse inoculation or feeding trials. The toxins can be typed with neutralization tests in mice. In toxico-infectious botulism, the organism can be cultured from tissues. On egg yolk medium, toxin-producing colonies usually display surface iridescence that extends beyond the colony. Although the vegetative form of the bacteria is destroyed by boiling, the spore itself is not killed by the temperatures reached with normal sea level pressure boiling, leaving it free to grow and again produce the toxin when conditions are right. A recommended prevention measure for infant botulism is to avoid giving honey to infants less than 12 months of age, as botulinum spores are often present. In older children and adults, the normal intestinal bacteria suppress development of C. botulinum. Prognosis. Epidemiology. United States. While commercially canned goods are required to undergo a botulinum cook in a pressure cooker at 121 degrees Celsius for 3 minutes, and thus rarely cause botulism, there have been notable exceptions. Two were the 1978 Alaskan salmon outbreak and the 2007 Castleberry's Food Company outbreak. Foodborne botulism is the rarest form though, accounting for only around 15% of cases and has more frequently been from home-canned foods with low acid content, such as carrot juice, asparagus, green beans, beets, and corn. However, outbreaks of botulism have resulted from more unusual sources. In July 2002, 14 Alaskans ate muddock from a beached whale, and 8 of them developed symptoms of botulism, 2 of them requiring mechanical ventilation. Other, much rarer sources of infection include garlic or herbs stored covered in oil without acidification, chili peppers, improperly handled baked potatoes wrapped in aluminum foil, tomatoes, and home-canned or fermented fish. When canning or preserving food at home, attention should be paid to hygiene, pressure, temperature, refrigeration, and storage. When making home preserves, only acidic fruits such as apples, pears, stone fruits, and berries should be bottled. Tropical fruit and tomatoes are low in acidity and must have some acidity added before they are bottled. Oils infused with fresh garlic or herbs should be acidified and refrigerated. Potatoes which have been baked while wrapped in aluminum foil should be kept hot until served or refrigerated. Because the botulism toxin is destroyed by high temperatures, home canned foods are best boiled for 10 minutes before eating. Metal cans containing food in which bacteria, possibly botulinum, are growing may bulge outwards due to gas production from bacterial growth. Such cans should be discarded. Any container of food which has been heat treated and then assumed to be airtight which shows signs of not being so, e.g., metal cans with pinprick holes from rust or mechanical damage, should be discarded. Contamination of a canned food solely with C. botulinum may not cause any visual defects. Only sufficient thermal processing during production should be used as a food safety control. United Kingdom. There is a vaccine, but its usefulness is unclear as it is associated with significant adverse effects. As of 2013, there are efforts ongoing to develop a better vaccine. Botulism is generally treated with botulism antitoxin and supportive care. Supportive care for botulism includes monitoring of respiratory function. Respiratory failure due to paralysis may require mechanical ventilation for 2 to 8 weeks, plus intensive medical and nursing care. After this time, paralysis generally improves as new neuromuscular connections are formed. In some abnormal cases, physicians may try to remove contaminated food still in the digestive tract by inducing vomiting or using enemas. Wounds should be treated, usually surgically, to remove the source of the toxin-producing bacteria. In adults, botulism can be treated by passive immunization with a horse-derived antitoxin, which blocks the action of the toxin circulating in the blood. A trivalent antitoxin containing antibodies raised against botulinum toxin types A, B, and E is used most commonly. However, a heptavalent botulism antitoxin has also been developed and was approved by the US FDA in 2013. In infants, horse-derived antitoxin is sometimes avoided for fear of infants developing serum sickness or lasting hypersensitivity to horse-derived proteins. To avoid this, a human-derived antitoxin has been developed and approved by the US FDA in 2003 for the treatment of infant botulism. This human-derived antitoxin has been shown to be both safe and effective for the treatment of infant botulism. However, the danger of equine-derived antitoxin to infants has not been clearly established, and one study showed the equine-derived antitoxin to be both safe and effective for the treatment of infant botulism. Trivalent botulinum antitoxin is derived from equine sources utilizing whole antibodies. In the United States, this antitoxin is available from the local health department via the CDC. The second antitoxin, heptavalent botulinum antitoxin, is derived from despeciated equine IgG antibodies which have had the Fc portion cleaved off, leaving the F2 portions. This less immunogenic antitoxin is effective against all known strains of botulism where not contraindicated. The paralysis caused by botulism can persist for 2 to 8 weeks, during which supportive care and ventilation may be necessary to keep the person alive. Botulism is fatal in 5% to 10% of people who are affected. However, if left untreated, botulism is fatal in 40% to 50% of cases. Infant botulism typically has no long-term side effects but can be complicated by treatment-associated adverse events. The case fatality rate is less than 1% for hospitalized babies. Globally, botulism is fairly rare, with approximately 1,000 cases yearly. In the United States, an average of 145 cases are reported each year. Of these, roughly 65% are infant botulism, 20% are wound botulism, and 15% are foodborne. Infant botulism is predominantly sporadic and not associated with epidemics, but great geographic variability exists. From 1974 to 1996, for example, 47% of all infant botulism cases reported in the U.S. occurred in California, China, Canada. Between 1990 and 2000, the Centers for Disease Control and Prevention reported 263 individual foodborne cases from 160 botulism events in the United States with a case fatality rate of 4%. 39% occurred in Alaska, all of which were attributable to traditional Alaska Aboriginal foods. In the lower 49 states, home canned food was implicated in 70 events, with canned asparagus being the most numerous cause. 2 restaurant-associated outbreaks affected 25 persons. The median number of cases per year was 23, the median number of events per year was 14. The highest incidence rates occurred in Alaska, Idaho, Washington, and Oregon. All other states had an incidence rate of 1 case per 10 million people or less. The number of cases of foodborne and infant botulism has changed little in recent years, but wound botulism has increased because of the use of black tar heroin, especially in California. Ukraine. All data regarding botulism antitoxin releases and laboratory confirmation of cases in the U.S. are recorded annually by the Centers for Disease Control and Prevention and published on their website. Other species. The largest recorded outbreak of foodborne botulism in the United Kingdom occurred in June 1989. A total of 27 patients were affected. 1 patient died. 25 of the patients had eaten 1 brand of hazelnut yogurt in the week before the onset of symptoms. Control measures included the cessation of all yogurt production by the implicated producer, the withdrawal of the firm's yogurts from sale, the recall of cans of the hazelnut conserve, and advice to the general public to avoid the consumption of all hazelnut yogurts. From 1958–1983 there were 986 outbreaks of botulism in China involving 4,377 people with 548 deaths. From 1985 to 2015 there was an outbreak of 91 confirmed cases of foodborne botulism in Canada, 85% of which were in Alaska Native communities, especially the Inuit of Nunavut and First Nations of the coast of British Columbia, from eating traditionally prepared marine mammal and fish products. In 2017, there were 70 cases of botulism with 8 deaths in Ukraine. The previous year there were 115 cases with 12 deaths. Botulism can occur in many vertebrates and invertebrates. Botulism has been reported in rats, mice, chicken, frogs, toads, goldfish, aplysia, squid, crayfish, Drosophila, leeches, etc. Death from botulism is common in waterfowl. An estimated 10,000 to 100,000 birds die of botulism annually. The disease is commonly called limberneck. In some large outbreaks, 1 million or more birds may die. Ducks appear to be affected most often. An enzootic form of duck botulism in western USA and Canada is known as western duck sickness. Botulism also affects commercially raised poultry. In chickens, the mortality rate varies from a few birds to 40% of the flock. Botulism seems to be relatively uncommon in domestic mammals. However, in some parts of the world, epidemics with up to 65% mortality are seen in cattle. The prognosis is poor in large animals that are recumbent. In cattle, the symptoms may include drooling, restlessness, uncoordination, urine retention, dysphagia, and sternal recumbency. Laterally recumbent animals are usually very close to death. In sheep, the symptoms may include drooling, a serous nasal discharge, stiffness, and incoordination. Abdominal respiration may be observed and the tail may switch on the side. As the disease progresses, the limbs may become paralyzed and death may occur. Phosphorus-deficient cattle, especially in southern Africa, are inclined to ingest bones and carrion containing clostridial toxins and consequently suffer lame sickness or Lahmsjucht. A recent study has demonstrated an effective vaccine against cattle botulism associated with Clostridium botulinum serotypes C and D. The clinical signs in horses are similar to cattle. The muscle paralysis is progressive. It usually begins at the hindquarters and gradually moves to the front limbs, neck, and head. Death generally occurs 24 to 72 hours after initial symptoms and results from respiratory paralysis. Some foals are found dead without other clinical signs. Clostridium botulinum type C toxin has been incriminated as the cause of grass sickness, a condition in horses which occurs in rainy and hot summers in northern Europe. The main symptom is pharynx paralysis. Domestic dogs may develop systemic toxemia after consuming C. botulinum type C exotoxin or spores within bird carcasses or other infected meat but are generally resistant to the more severe effects of Clostridium botulinum type C. Symptoms include flaccid muscle paralysis. Dogs with breathing difficulties will require more intensive care monitoring. Muscle paralysis can lead to death due to cardiac and respiratory arrest. Pigs are relatively resistant to botulism. Reported symptoms include anorexia, refusal to drink, vomiting, pupillary dilation, and muscle paralysis. In poultry and wild birds, flaccid paralysis is usually seen in the legs, wings, neck and eyelids. Broiler chickens with the toxico-infectious form may also have diarrhea with excess urates.